Hi, my name is Alyssa Yuvienko, and this presentation is for extra credit for Dr. Gurley's class, and it is about the foxp2 gene. And here is a tertiary structure of the foxp2 gene and where it is located on chromosome 7. For the first part of the introduction, I wanted to begin with a general overview of the foxp2 gene. The foxp2 gene is shown to be genetically linked to the development of language and is hence dubbed as the language gene. Research has shown that the foxp2 gene is not unique to humans, but can be found in many species from chimpanzees into mice. However, the foxp2 gene that humans carry is distinct in its specific version or its mutation. Shown to the right here is an annotated phylogeny diagram that shows how humans are distinguished from mice and chimpanzees and other non-human primates. And we can see here that it is only by two non-synonymous mutations, as we can see as indicated in blue in the legend. Since it is only by two non-synonymous mutations, this indicates that this gene was highly selected for. This gene is crucial for the development and function of different parts of brain circuits in the neocortex, in the basal ganglia and cerebellum, and other language centers of the brain. And mutation of the foxp2 gene can cause speech and language disorder that also provides insight into the neurobiology of language. An example is a point mutation that alters the foxp2 transcription factor, which may lead to linguistic and grammatical impairment through some cited examples, such as developmental verbal dyspraxia or childhood expressia of speech, as well as issues with written language. Since the foxp2 gene is involved in the regulation of certain gene expression, we know that more in-depth regulatory mechanisms need to be further studied to get a better insight on the foxp2 gene and its functions. For the second part of the introduction, we will go over some previous research on the foxp2 gene. And as we stated before, modern humans, they are defined by the foxp2 gene by two amino acid alleles. And in 2007, a study by Cross stated that Neanderthals also carried those two derived amino acid alleles. It has been estimated that the branch for the foxp2 allele had been selected for nearly 300,000 years ago. And in 2009, a study by Fisher showed that reduced foxp2 dosage yields some interesting things in different organisms, such as abnormal synaptic plasticity in the brain and impaired motor skill learning in mice. And it also disrupts vocal learning in songbirds. And this study was done in 2009. And also, heterozygous foxp2 mutations have been known to cause developmental verbal dysplasia, and foxp2 downregulates the CNTNAP2 expression, which is a gene which represents a promising candidate for the involvement in language impairments. And this provides a key example to the foxp2's gene regulatory factor. And also another relevant point to this presentation is that the foxp2 gene shows similar neural expression patterns in humans, monkeys, mice, rats, birds, in different neural circuits, so they are in different areas of the brain. A recent article published in August of 2018 in the journal Cell actually questions the evolutionary significance of the foxp2 gene. This review article stated that they did not find evidence of the recent or positive balancing selection at the foxp2. However, they did find that there was an intronic region that was enriched for highly conserved sites among humans. Also, the study stated that there was no evidence for recent selection of the foxp2 gene across diverse populations, which makes it known that there needs to be a new hypothesis for language evolution, such as a gene regulatory mechanism. Also, another issue about the foxp2 gene's old research was that the exon 7 variants are not human-specific, like they stated before, because Neanderthals and Denisovans and anatomically modern humans, they both had the same allele, same two alleles that had originated greater than 600 kiloyears ago. So as I stated before, the intronic region of the foxp2 is enriched for human-specific SNPs. And that intron is located in the diagram right here, and this needs to be further analyzed and studied, the significance of the function of that intronic region for human evolution today. With the given background information, next is the presentation topic. The topic of this presentation is the association of linguistic and grammatical impairment and genetic variants of the foxp2 gene in non-human primates. 3 research articles will be presented in this presentation that shows studies supporting the impact of the foxp2 variants against different organisms using a variety of experimental methods, which include, but are not limited to, sound analysis, qPCR expression analysis, cell cultures, and even publicly available whole genome data. In this presentation, we'll individually break down each research report and its experimental methods and results. The title of the first research article is foxp2 variation in great ape populations offers insight into the evolution of communication skills. And this was published in December of 2017 in the journal Nature of the Scientific Reports. And the first author of the study was Nikki Stays. And the aim of the study was to analyze the foxp2 coding variation in a sample of great apes. The researchers further sought to investigate the allelic length variation in 2 poly-Q tracks located in exons 5 and 6 in the foxp2 gene of non-human primates. And the hypothesis based off of previous research was that the results would provide evidence of regulatory changes that are unique to the population of great apes and will provide insight onto the evolution of language in modern-day humans. There was a lot of different experimental strategies for this research article because there was a lot of different types of data. Firstly, the researchers maximized their sample size by including data from a preliminary study. Then genotyping was performed using public genomic data for chimpanzees, bonobos, gorillas, and orangutans, and then mapped out using a BWA program. And then SNPs were recalled using the GATK Unified Genotyper, and then another program called Genesis. Then after that, the coding exons were amplified by PCR analysis and then Sanger sequenced. And then lastly, a microsatellite analysis of the polyglutamine tracks were performed. Figure 2 here showed a comparison between species of the length of their polyQ tracks. And this alignment was taken by use of subspecies ascertainment, DNA extraction, and PCR analysis, and also, as well as DNA sequencing. The significance of the polyQ tracks is that they are encoded by a mixture of CAG and CAA codons that are often repeated in tandem, and these types of repeats have a higher mutation rate and can serve as a functional modulator of eukaryotic mutations. Also, polyQ track variation has been correlated to show an impact on gene expression by regulating gradients as a turning knob effect. It is dubbed the turning knob effect, as shown with the RUNX2 transcription factor, which would completely alter the structure and function of the gene. So as we look at the diagram, we can see all the different species listed to the left, and we can see that it's 10 different ones. The two polyglutamine stretches shown here are shaded, and they show repeated times in tandem positions of the polyQ gene. And the dots indicate similarity to the previous sequence. And also with the species within species, when we're comparing two different species together, there is an A that's marked for ancestral and a D which is marked as derived. So here you can see that in chimpanzees and orangutan to distinguish between the two species. These results are significant because it is reported here that there are little variation between humans, but the study shows that orangutans and chimpanzees have a higher genetic diversity. The next figure we will be looking at from the first research article is the figures 3A and 3B because they go together. And figure 3A is a representation of the foxp2 gene, and the boxes represent exons and the lines here represent introns. So boxes are exons and the lines are introns. And the translated exons are shown in black here. Also here is showing the domains of the foxp2 gene that is being coded for, and here we can see the two polyglutamine tracks, which are indicated by the two hues, and a zinc finger motif, and also the forkhead domain as indicated with fox, and an acidic T-terminus. Also here we can see the start codons as indicated in ATGs and our stop codon, which is indicated with the TGA. And in here, represented in here, is the P626T mutation, and it's zooming in on the P626T mutation found in orangutans, and it is a heat map, and the redder it gets is the more chances that it's going to be a mutation, and the more neutral it is, the more is indicated in blue, the more blue it will be. So here we can see that it has a very likely chance of mutation. The second research article's title is called "A foxp2 mutation implicated in human speech deficits alters sequencing of ultrasonic vocalizations in adult male mice." And this journal was published in 2016, and the name of the journal is Frontiers in Molecular Neuroscience. The first author of this article is Martin Becker, and the aim of the study was to analyze adult male mice carrying the KE mutation using ultrasonic variations and statistical methods. And the hypothesis of the study is that ultrasonic vocalizations and advanced statistical tools will show variation of KE heterozygous male mice in accordance with a different environmental stimuli. And for the overall experimental strategy, mice were first genotyped by polymerase chain reaction using key primers. Vocalization were then recorded with an special ultrasound microphones from 10 heterozygous and 9 wild-type male mice around 8 to 9 weeks of age. And then female mice were placed in cage for stimulus and acoustic analysis, syntax analysis, and oxygen and carbon dioxide, respiratory air were measured. And after recording sessions, 6 heterozygous and 6 wild-type male mice were used to trace connections between the laryngeal muscles and the primary motor cortex of the brain. Here we have figure 1D and figure 2B. They are on the slide together because figure 1D explains components of figure 2B. And starting with figure 1D, this shows the sonogram for the 4 syllables being analyzed, which is S, D, U, and M. And here you can see that it shows the frequency in kilohertz on the y-axis and the second in time of in milliseconds on the x-axis. And these datas are translated into colors on figure 2B. Here you can see the S, D, U, and M in different various colors. And here on the x-axis, or on the horizontal component here, you can see the different environments that the researchers put the male mice into. And this was done with the heterogeneous mutant mice, and the 4 environments are listed below. It's the female with urine, and a live female, and an anesthetized female, and an anesthetized male. And here we can see the syllable repertoire composition, where it seems that the S syllable is dominated all across the board for the foxp2 syllable expression. Heterozygous mutations in the KE family causes speech apraxia, which is characterized in difficulty in producing complex sounds. Figure 4C to the right gives an example of a complex syllable sequence, and these are constructed by frequencies and multiple pitch jumps. And the graphed y-axis on the left indicates the ratio of complex syllables to simple syllables. But more specifically, the graph is measuring the number of sequences with 2 or more M syllables divided by the sounds with only 1 or no M syllables in each stimulation context. So here was the ratio of the complex to the simple sequences, and here are the different environmental stimuli. They had a sample of female, the males going into the female urine stimulus, and then the males being introduced into with a live female, or an anesthetized female, or an anesthetized male. Gotta do that again. And here we can clearly see that the live female gave the most stimulus for the foxp2 gene, or the ratio of complex to simple sequences. So in summary, the results showed that contributions of foxp2 to sequencing of vocalizations are conserved with humans, and they compared 4 major categories in this study for the acoustics. It was simple, up, down, and multiple pitch joints from wild-type to heterozygous male mice. And the foxp2 heterozygotes produced less complex sequences than wild-type mice. And also, there were a lot of unaccounted variables including different courtship songs during different seasons, or other unmeasured stimulus or variables. For the third research article, the title is Mapping of Human foxp2 Enhancers Reveals Complex Regulation. And this journal was published in 2018, and the title of the journal is Frontiers in Molecular Neuroscience. The first author of the study was Martin Becker, and the aim was that the researchers in this study sought to determine cell lines and promoters that are involved in the endogenous expression of the foxp2 gene. And the hypothesis of the study was that genetic mapping may lead to correlations that contribute to specific neural and behavioral phenotypes. For the experimental strategy, the foxp2 expression analysis occurred through qPCR, and it underwent immunoprecipitation of chromatin and digestion of chromatin through a modified 3C protocol. Then sequences underwent luciferase reporter assays. So first, for this research study, we're going to take a look at figure 1, and this data was extracted through qPCR analysis. And here on the x-axis, you can see the cell lines that are being measured against the relative endogenous expression of the foxp2 gene in these human cell lines. And the expression level of the HEK293 cell line were used as a reference, and differing from the HEK293 cell line were the SNKMC and the PFSK1 cells, and those were neuron derived. And as you can see, only those 3 cell lines were expressing the foxp2 gene, and the PFSK1 here on the graph shows almost an 80% decrease of expression than the HEK293 and SKNMC cell lines. This figure 4D hinges on the last figure 1, because it takes the data the data of the 4 neuroblast cell lines, the Kelly, the IMR32, and the SHSY5Y, and the SKNAS neuroblast cell lines that did not detect the foxp2 gene expression. And so these were taken and measured through chromatin conformation capture, or 3C analysis. And as you can see here, that there was enrichment. It shows it as and it diagrams it as distance from the anchor point in kilobases, and the units are in relative enrichment. And the indication of enrichment of these genes, even though it's lower than those positive cells, it suggests that these cell lines still physically interact with the TSS1 or the promoter 1 in gene regulation. For the summary of the research articles data portion we just looked over, we found that the HEK293 and the SNKMC and the PFSK1 cell lines were the only ones that had positively expressed the foxp2 gene, but they did an enrichment test that targeted DNA fragments using qPCR, and the enrichment observed in negative cells were much weaker than positive cells, but they still had data that indicated that the cell lines were physically interacting with the TSS promoter, indicating that there is more complex regulation for the foxp2 gene that still needs to be researched. So in conclusion, the phenotype of mice, soundbirds, orangutans correlate with mutations of the foxp2 gene that can give insight of the human's version of the foxp2 gene. And the inability to produce complex syllables were shown in data for heterozygous mutation of the foxp2 in mice. And the foxp2 is highly regulated, as the data showed, with only a few cell lines showing a strong expression for the foxp2 gene, and unaccounted variables may include sample size, organism limitation, the neural networks of a mouse might be different than a human, and the mating cues and environmental and stimulus cues may have been misleading in the study. There are two major applications, I believe, to the foxp2 gene, and the first one is the evolutionary application, where the importance of the selection of the foxp2 gene in developing cognitive thought and abstract thinking and even the emergence of art are very interesting to study through an evolutionary perspective through the foxp2 gene. And another application of this study is clinical, because the regulatory networks that are provided in this study would allow for analysis of dysregulation or give a mechanistic view of certain processes in the human body that may be used in pharmaceuticals and drug therapy. For future studies, some interesting future studies would be ones that provide more of an evolutionary insight on the molecular and cellular foundations of human language. And also in many of the articles, they talked about the potential role of neuroplasticity in the vertebrate brain, which could be very helpful as it repairs brain damage. So, and another future study could be the continued analysis of gene regulatory networks, such as the regulations such as upstream and downstream pathways. And another discussion for future studies could be the synaptic plasticity variation of intronic and regulatory regions as defined in many of the studies, and also tissue-specific studies, because the foxp2 gene is not limited to just the brain.